perfectly in time with the 10 minutes delay. So I inform all the attendees that we shifted for 10 minutes the entire second part of the morning program. So I will introduce the next speaker.
Excuse me. I interrupt you because Rosanne Minguin finally is inside in the panelist because she asked to all panelists help to enter. There was a Zoom problem just for the other panelists that the problem has been solved. Thank you very much.
Okay, that was for the evening session or
yes,
not
yes,
for the morning
yes,
session?
Yes.
Okay.
But there was a problem with the email with the email address or something like this because Zoom was refusing to move to attend it to panelists. Okay, but has been solved. Excuse me.
Very good.
But all for all panelists and that the problem has been solved.
Perfect. Thank you, Almo. And so the next presenter is Marco Gamba with a talk and with a paper on the acoustic diversity and complexity of the Maromizaha Forest Reserve in Madagascar. So if you, Marco, are with us.
Yes, I think, can you hear me now?
Perfectly, yes.
All right, so let's see whether I can share my
I
screen.
remember if you have audio samples.
Yeah, I did. I think I did everything as suggested, and now let's see whether we can do that. It's asking me for some password, so and let's see what happens. Okay, can you see my full screen slide now?
Yes.
All right, good. Cool. Thank you a lot for having me in for this presentation. I will talk about the acoustic diversity and complexity of the Maromizaha Forest Reserve in Madagascar. As you see from the long list of names, I got a lot of colleagues and collaborators that happily joined me in this long-term work. Let me start with some picture of Madagascar. Of course, Madagascar is a wonderful place where we got many different landscapes, different climates, different soundscapes. And Madagascar is also home to a number of endemic species, including more than 100 species of lemurs, but we'll come to lemurs later on in my presentation. I will take you to this special place in Madagascar called the Hankeni-Heni-Zahamena corridor, and in particular I'll focus on this area, the Andasibe-Mantadia National Park and its surroundings that we can see over here. These four forests you can see here are critically important areas for the study of many lemur species and in general for tropical rainforest environments. And in one of these forests, this one, Maromizaha, we got a little research center, and you may have heard of Maromizaha and its soundscape because 3 years ago the New York Times listed Maromizaha among the places whose soundscape is worth listening to, and I know that some of you are familiar also with the other places listed over here. So what we've done, we put a number of audio modes in several locations in the forest of Maromizaha and across two different areas. This area, which is the ecotourism area, and this other area, which is the research pristine area. In the ecotourism area, the environment, the forest environment is a bit more degraded and lower quality, while in the other area, the research pristine area, the forest quality is higher. We did our recording using 10 minutes recording over every 30 minutes and for 24 hours, and the results I'm presenting over here are related to a sampling of 150 days. The study was twofold. On one hand, we wanted to investigate the sound of the forest environment across the two different sites. And on the other hand, we wanted to see how the behavior of a target species was some way potentially changing according to the sound of the forest environments in the two different sites. I put these slides just to mention that we know where we want to go, but to some extent the data I'm presenting today are preliminary because we know we need more sites, more sampling days, and more sampling days, and of course more data about the animals of the different sites. So we did basically two things in this first part of the results I'm presenting. We calculated the acoustic indices for each site, compared them across the ecotourism area in red and the research pristine area in blue and green, and we extracted the features of these soundscapes using the SETI et al. 2020 PNIS procedure, and quantified the results using UMAP and random forest classifiers. So let's start with the first batch of results. But before starting with the quantitative data, I want to take you to Bevorundruru and have a look at the soundscape of one day in Bevorundruru. I'm not letting you listen to what happened over here, but we see a lot of things over here. We see birds, we see insects, we see a strong storm with rain and wind in the afternoon, and we come back to a quieter situation over evening and night. And we'll come back to this one later on. Let me provide you a very short guidance about what you'll see in the next graphs. You'll see the ecotouristic results over here. This is the red color denotes the ecotourism area, and the bluish and the greenish code is related to the research and pristine area. And above you'll see the results about the day, and below the results about the nights. And so let's start with the H index. So we have a lot of variation for the H index, but we see that there's no clear separation for the results we got during days or nights for the two areas, and the three sites of the ecotourism area and the other sites of the research area. And so we had a look at the variation of the index during the daytime, so average across the daytime, and see what was and the nighttime, and seeing what was happening. And of course you'll see a segregation of the reddish line paths from the greenish and blue paths if we got a strong difference in between the two, but if there's no strong difference between the ecotourism area and the research area, you will not see this separation. And this is what happened. So you'll not see a separation in between the red lines and the bluish and greenish lines. You see there is a very low red line over here, but there is even a lower line for the for one of the sites in the research pristine area, which has, as we said before, a higher forest quality. Then we calculated a normalized acoustic diversity index. Again, there's quite a lot of variation. There's no clear difference in between the two sites. Of course, I tested this difference, and this difference were not statistically significant. And when we look at the, let's say, day variation across the different sites, again we don't see a clear separation. Here we got a very low situation during the central times of the day for one of the red like the ecotourism areas here, but we see a lot of variation even in the other values, and there's no clear difference. And if we look at the top, one of the top areas in terms of acoustic diversity is one of the areas that is in the ecotourism area. Same stuff for the normalized NDSI. So we see that there is again a lot of variation. We see that some of the very top sites are those in the research and pristine area, which is some way what we expected, but again we see a lot of variation for the other sites. And so again, it seems that there's no catching of this difference in the environmental quality in between the indices that I'm presenting to you today. And I can tell you that we tested a lot of other indices, and again we haven't found the clear differences, and so we can discuss this later. And again, for the NDSI, we did the variation across the day, and you see that it's more or less similar to what we seen before. So we got A values for some of the areas in research pristine part of the forest, but we got a lot of variation also on those sites, and some of those are clearly overlapping with the values coming from the ecotourism area, which is more impacted. Then we did a UMAP visualization, as I said before. I got to say that the feature extraction of SETI and colleagues is a very nice procedure, a very nice pipeline of analysis. I interrupted that pipeline because I think that you got a number of strange parameters that I was not very convinced to use for the visualization. So I took out all those with a very low standard deviation, and I did a PCA before the UMAP visualization, and this is what we got. And when we run a random forest classifier on these, actually we can classify more or less 85% of the sites to their area. So there is some way emerging a difference between the forest which higher quality compared to the forest with lower quality, and this 85% is pretty much equilibrated in between the two. So it's 84 for one and 85 for the other, so it's pretty much the same. Then let me change the spotlight to a target species that we are studying for many years, and coming back to the spectrogram that I showed before. All what happened over years is related to these species that I want to introduce to you right now. And so let's focus on that little portion of the spectrogram and on these species. Indri is a flag species for the conservation of Malagasy lemurs, a unique species for several behavioral and biological traits. And they live in groups that we started studying many years ago with Christina Giacomo and other colleagues. 17 years now we are spending studying these species in Madagascar, and we wanted to develop a quantitative approach on many aspects of their biology, and now we know that they live in groups of 2 to 6 animals, and they are a pair living species. And so we can say, after we did some genetic analysis, that these groups are family groups. But what's great about these species is that they sing, and this is a very special thing about the Indris, because there is a very small number of prima species that are providing songs, and so we really wanted to understand the function of these songs. And across the years we understood that each social group occupies and actively defend an exclusive and stable territory, and they use songs to advertise its occupancy. And so songs are a key method in keeping the spacing in between the neighboring group in the forest. And I think that some of you are for sure familiar with the songs, but for the others, I will try to play a video, and I hope you will be able to hear the audio. Remember that in the video you'll see an animal singing. The song is 99% of the times a chorus, so you got the different animals singing at the same time. We'll focus with the video on one of these animals, but you can hear the other singing in the background. Let's look at this. Maybe. portion of the song is the one in which the units are given in descending frequency. They are called descending phrases that you probably read about in many of our publications. So, what we learn over the years about these songs, the songs are highly sexually dimorphic, that adult flexibility is present, especially in females, and that genetic correlates with song traits, but mainly in males. And we also know from a recent paper we published that songs are changed during ontogeny. So there was some potential to imagine that, let's say, a group can adapt their song to the forest environment if it's the case. And we wanted to do that and to test that. So we took the groups living in the ecotourism area and the groups living in the other area, the pristine research forest over here, and we recorded these songs with traditional recorders, not with the audio mode or the SM4, but using sound device and a Sennheiser directional microphone. It's a very important step in this process, and you get why in my next slides, is that we recognized each individual individually. And so we identified the singer at a particular point, or the singers at a particular point in the choruses, and we've been able to split the song in the different contribution of the different individual participating to the song. This is a key point because we were able to quantify the overall duration, but also the overall phonation of a single individual during the song. And for each unit of each individual contribution, we took other parameters, the frequency range, duration, max frequency, and low frequency. Coming to the results, we were testing different across the two different areas. As I said before, we used GLMM to control for the group and for the individual, and we found no significant difference for the overall duration, no significant difference for the overall phonation, and no significant difference for unit duration or for frequency range of the unit, and also no significant difference for the maximum and the minimum frequency. But let me say that the maximum and minimum frequency shows, let's say, a marginal, let's say, some way a tendency towards significance. So let's go into a quick wrap up because time is flying. We see that acoustic indices did not catch the difference in the two areas that we wanted to compare, and maybe a combination of indices, a larger sampling period, or something else that I cannot tell you why, can be useful to compare the two areas. But it's also possible that there is an effect of the canopy. Sylvan talk a couple of days ago was pointing, in my opinion, to a very good direction. It's difficult to quantify whether the dense canopy of the high quality forest is some way attenuating the sound coming to the ARUs, and so this can be also a reason for the lack of differences. It's also true that we found some difference emerging for an MFCC approach, but these differences that we found at the end in the two forest soundscapes is not reflected with the INDRIS song that we recorded. And this can be for two reasons, or there is no acoustic adaptation, or there's no need for acoustic adaptation because the song works well in both environments and travels well a distance, or maybe because they cannot adapt. And we don't know that, but we promise we'll investigate in the future. Thanks to all my collaborators. You can see their faces right now, and thanks if you want to also, if you're interested in animal behavior, you want to check our free software, you got an address over here. And thanks for your attention.
So thank you very much, Marco, for your very clear presentation and very moving video. Actually,
thanks.
Was it a mating song, or what is it? Was it
just a territory? Yeah, it's an advertisement song. It's given when they are in the middle of their territory and they want to advertise prioritize where they are, and we know that there but might be different function for that, but they have another song when they clash in between neighboring groups, and they really want to physically defend the territory, and they have another song when they want to regroup, and they got lost in between different activities like feeding or moving in the forest, and they got lost in between members of the same family group, and they want to regroup. There is another type of song. So for sure, this some way signal that songs are working very well in that environment, probably not depending on the fine traits of this singing.
I mean, just a comment before passing to the questions and answers. These glissandos, these portamentos, it's a very clever way that they developed to be heard, because portamentos can be, can be very heard much better in a reverberant environment like a rainforest. Yeah. And so that's. But they are so sad. It's beautiful. It's just beautiful.
When you listen to these songs in the forest, it's really an experience. Something you can feel. You can feel with your body. It's really a great experience to have the chance to hear the song in the forest with the mist and everything. It's really
with the reverberation, many songs and from many places.
Yes, it's contagious, and you see the song moving across the forest. It will be a great thing to capture with your project.
Let's do that.
Take a note. Take a note.
So I see Jerome has a question.
Yes, even to just to go on what just David suggested, maybe you could find a signature in the frequency modulation of the sounds. That could be there.
And we studied some way. Sorry. Let's go.
Yeah, no, please.
No, we've been looking at this frequency modulation profile of the different songs, and we also some way suggested some let's say degree of variation in between females and males that can be different in one of the papers we published last year. I think that there is more to do, of course. It's not necessarily something that will show a difference in between the group living in the different forests, in my opinion. But let's see what happens.
Okay, thank you. And so another comment, it's beautiful place, beautiful sounds, beautiful research, but it's a remark I've already done. Using the acoustic indices, I think that there is something that we might not do in the best way, because what we did it there. So we use you test different indices, and you try then to see if they work properly according to an hypothesis, which is a strong hypothesis here, which is that you should have a lower diversity in the ecotourism area than in the pristine one. But I think that actually you check the validity of the indices downstream when you should test it upstream. So that means that you should test the indices before comparing them with the ground truth. So at some point, and that's again the very difficult thing, is to have someone who annotates the files, an expert, and so you can compare manual annotation and what the acoustic indices do. And then you select the best index or the best indices or combination of indices, and then you apply it on your two different sites or whatever. And then you can use them with some confidence.
I totally agree with you. Yeah, this is something that some way we started doing. Of course, as you know, there might be also an effect of other factors that we've not been checking over here. For example, we've not been checking for rainy days, and we are in the rainforest, and so this can be a difference, of course, or can be something that is masking the difference. And so, of course, we got to do in a deeper detail about that, and yes, I think this can be one of the reasons. I also really think that the kind of distance, I mean, you remember I wrote you an email years ago asking about distance at which you can detect things with the different recorders, and this can be also something that of course changes across the different force qualities, and this can be something that can be taken into account. Yeah.
That's a technical point that Sylvain might answer, but the thing is more in the way we process the science. So
of course.
we should not, and we did it, so we did that mistake. We should not check the validity of the indices upstream, but downstream, but upstream. Because you can't check something according to an hypothesis. You don't know if there is more or less diversity in your ecotourism and not ecotourism. Or maybe you have it with other data like survey or
now we have this data.
Okay, so that's something you should mention, maybe.
I'm answering one of the questions that I can read over here, which is whether human activity affects the indices, but it's not, because it's not included in the recordings that we've done. So I think that this is not something affecting the results I showed now, but of course in general it can happen. Thank you.
Okay, I must I must say that we are delayed. So thank you, Marco Gamba, for your
thank you so much
great presentation.